The overall goal of this experiment is to detect and track non-apoptotic caspase activity in live animals using Drosophila melanogaster as a model.
Caspases are proteases that cleave many cellular components to cause apoptotic cell death. Therefore, the presence of active caspases in cells is often assumed to reflect impending cell death. However, growing evidence indicates that the same caspases that cause cell death also have normal roles in healthy cells. And these day jobs of caspases include regulation of neuronal activity, mitochondrial functions, even the suppression of alternative cell death pathways. There are two major difficulties in studying the day jobs of caspases. First, normal basal levels of caspase activity are typically below the limits of detection by available technologies. And secondly, even when basal caspase activity is detectable, it is difficult to distinguish from the caspase activity that will lead to cell death in the near future.
To solve this problem, we developed CaspaseTracker, a more sensitive caspase biosensor that can permanently label cells that have survived caspase activation. This biosensor will enable us to detect and track the cells with non-apoptotic caspase activities and to study its functions in live animals.
The Drosophila in vivo CaspaseTracker biosensor system consists of two components. The caspase